Absolutely. So for the next speaker, we have Victoria Cairoli from the Instituto Multidisciplinario de Investigaciones en Patología. Yes, are you there, Victoria, in the crowd?
Yes. Hi. Can you hear me?
Excellent. Yes, and I can see you. So you should be able to share your screen.
Okay.
Yep, we could see it.
You can see my presentation?
Bravo. Yep, excellent. So please go ahead, introduce yourself, and we look forward to hearing your work.
Okay, thank you very much. Hello, everyone. My name is Victoria Cairoli, and I'm going to present the work entitled microRNAs from plasmatic extracellular vesicles as biomarkers of liver fibrosis in cases with chronic hepatitis C, which is a part of my doctoral thesis. So, as an introduction, so let me see here. The hepatitis C virus is one of the main causes of chronic liver disease because it causes chronic hepatitis C in most patients that is characterized by cell damage, inflammation, and fibrosis that in the worst case scenario could lead to cirrhosis or hepatocellular carcinoma. So the staging of liver fibrosis is fundamental in the clinical practice, and currently the gold standard method to do so is by liver biopsy. However, it is a very risky procedure and it's very invasive and it's very expensive. So the search for new, less invasive biomarkers is urgent. So in this context, extracellular vesicles have gained a lot of popularity in the past few years as part of liquid biopsies because they are very stable and because of their content. As you know, extracellular vesicles carry all types of molecules. And in this work, we are going to focus on microRNAs, which are known for regulating transcription by binding to the messenger RNA. So based on the hypothesis that microRNAs may modulate fibrogenic signaling pathways in the liver, we asked the question if they could serve as biomarker for assessing liver damage. So the aim of our study was to characterize the microRNA signature of plasma-derived EVs with respect to the severity of liver fibrosis and to evaluate their diagnostic accuracy to discriminate liver damage stages. In order to achieve this aim, we enrolled 50 adult patients with chronic hepatitis C, and we assessed liver fibrosis by means of the Metabir index, which classifies patients with significant fibrosis or non-significant fibrosis. 36% of our cohort has significant fibrosis, and we obtained blood samples from all of them. We separated the plasma and we proceed with a clarification step with multiple centrifugations, a dilution in PBS, and a final filtration step. We then isolated the EVs with a commercial kit and then proceed with the characterization of the EVs, which was made by two techniques, electron microscopy and nanoparticle tracking analysis. And then we proceed with the RNA extraction with the same kit that I previously mentioned. So then we proceed with the massive sequencing that was performed with the library preparation was with a specific kit for small RNAs, and it was carried out on an Illumina platform. The final step was the bioinformatic analysis to identify the microRNAs, to identify those that were differentially expressed between fibrosis stages group and to determine the diagnostic accuracy of those microRNAs. For the results, the EV characterization by transmission and cryo-electron microscopy, we found spherical bilayer particles with a mean diameter of 130 nanometers that were compatible with EVs, and by nanoparticle tracking analysis, no significant differences in EV size or concentration were found between liver fibrosis stages. The differential expression analysis results show, as we can here see in the volcano plot, that we found two dysregulated microRNAs we found the upregulation of MIR122 and the downregulation of MIR92. Interestingly, MIR122 is known to promote HCV replication, and MIR92 was described to be downregulated in hepatocellular carcinoma, promoting cancer-associated pathways. Finally, we studied the diagnostic performance of these two microRNAs, and we found that they had moderate diagnostic value to discriminate significant fibrosis because they showed an outlook between 0.7 and 0.8, which is not great. So with these results, we perform a logistic regression analysis in order to generate a score that combines both microRNAs and see if this score could better discriminate the patients with significant fibrosis. And we found that this new score had an outlook greater than 0.8, which means that it had an excellent diagnostic accuracy for discriminating patients with significant fibrosis. So the conclusion of this work could be that chronic hepatitis C patients with different stages of liver fibrosis present a specific EV-derived miRNA fingerprint, and that this specific signature could allow the use of these microRNAs derived from the EVs as biomarkers of unfavorable progression, and that they could be used in a diagnostic panel. Thank you very much for your attention, and if you have any further questions, please do not hesitate to ask.
Yeah, excellent talk, Victoria. That was brilliant. So I'm fascinated by this work, especially from a liquid biopsy angle that can be implemented in the clinic. Two questions. So your experimental design, the two cohorts, what was the difference between them again?
It was a cohort with chronic hepatitis C, and we had patients that were only infected with the hepatitis C virus, and we had patients that were co-infected with HIV virus.
Okay, so yeah, so then my two questions then based on that are, do you have a normal control? You know,
no,
what do these what do these microRNAs, their expression levels look like in normal controls? And then two, my second question is in the slide before this, you do excellent statistical work, your logistic regression, the ROC curves look incredible. It really does. How does it stack up to traditional biomarkers for hepatic fibrosis? Why look at liver EVs while why not look at albumin, bilirubin, ALT, AST? How does it stack up compared to those?
I'm going to answer first the second question because I forgot the first one, so later if you can repeat it. Now that, as I mentioned in the introduction, the gold standard method to determine liver fibrosis is liver biopsy. There are other methods, as you say, transaminases, other indexes like FIB4 and APRI, but they are not perfect. And for instance, these two indexes, APRI and FIB4, they are widely used in the clinical practice, but there is a gray area with these indexes that you have patients that cannot be classified with these indexes. So it is very controversial. Now there are other methods like images, FibroScan, that it is being used, but not every hospital has that kind of machines. For instance, I'm from Argentina, and here there are a lot of low-income cities that don't have the access to such technologies. So it is very urgent, the need for specific biomarkers that go with greater accuracy. I don't know if I answer your second question.
That made complete sense. Thank you so much.
And what was the first one?
The first one was about controls, the normal.
Yes. Yeah, we do not have controls. We have other work, but we had to choose which one was going to be presented here. We have other work in which we took the hepatitis C, the ones that were only infected with the hepatitis C virus, and used that as a control to see how it affected the presence of the second virus, the HIV. But as you know, massive sequencing is a very expensive technique, so we couldn't afford to make controls.
And any evidence of viral nucleic acids loaded in these EVs?
I think they are, but when we made the bioinformatic analysis, you have to match with a reference genome, and we did it with the human. So we only search for human and microRNAs.
Okay. Yeah, so if you change the reference data set you compare it to, then you might find some viral signatures in there as well. That would be interesting.
Sure. Yeah, sure.
Alex, would you like to make your question?
Okay. Hello. Can you hear me?
Yes.
Yes.
Okay. Very excellent research, Victoria. Very excellent research. So okay. I'm a bit curious about your isolation. I think these are viral vesicles, and coincidentally, I'm equally working on viral vesicles. So I would really like you to explain the isolation method again. I think you didn't really throw more light on the isolation method you used, because these are viral vesicles, and sometimes viral vesicles are very hard to isolate. So I really like you to explain a little bit about the isolation.
We use a commercial kit. We use this here. I'm going to. This kit is from the brand Cairoli, and it's based on columns that had a membrane, and you have to, well, these kits don't really tell you what's in the buffer, but you have to put the sample, the pre-clarified plasma, and then you do a lot of washes with different buffers, and with some specific buffer, the EVs are retained in the membrane, and with another buffer, you elute those EVs. But this is a kit, a commercial kit.
Okay, thank you. I think I would reach out to you privately to learn more about the kit. Thank you.
Okay, yeah, sure. Are we done?
So I think we can move on. Thank you, Victoria. I had a question, but we need to move on. Sorry.
Okay.
But maybe I can then ask you on Discord channel.
Yeah, sure.
Perfect. Thank you so much for your presentation. So yeah, I will share now